Bismillahirrahmanirrahim. Today we are just going to organization of nervous system, in which we try to introduce the nervous system, and it is not introduction for the physiology, also for the neuroanatomy and neurophysiology both. So organization of nervous system. What do you mean by nervous system? Which control our body. So at the top is the nervous system, and after the nervous system, your second important system is the endocrine, which regulate our homeostasis or our body system. So we are just going to what are the parts of the nervous system. So we start with there are two parts. The one is called central nervous system, the other is called peripheral. What do you mean by peripheral? Simple. Peripheral mean what? Means something distal. So central means something at the center. So if you consider the central one, so what is included? Brain and your spinal cord. So this is considered as the central. And what do you mean by peripheral? Mean nerves which are going to supply our throughout the body. So those, if they are coming from the skull region or your head and neck, they are considered as the cranial nerve. So if they are coming from your spinal cord, they are considered as the spinal nerves. So there are 31 pairs of spinal nerves. There are 12 pairs of your cranial nerve. So peripheral nerves, there are 31 pairs. So pair means they are coming on both sides, left and right. Okay. So ganglia and cranial and spinal. So if you consider the why we call them neuroanatomy, because it's related to neuron. So neuron study. So neuron has three parts. What are those? Cell body, axon, dendrite. So the collection of cell bodies is called ganglia. All right. So if this collection of cell bodies in the peripheral nervous system, so term is used ganglia. If same thing, the cell body collection is in your central nervous system, either in the brain or in the spinal cord, we call them the nucleus. All right. Nucleus and ganglia. So other thing is the autonomic system. So one is a somatic, which is under our control, and the autonomic means what? Involuntary. So if you see in this picture, so this is what? Brain, and here is what? Spinal cord. So this spinal cord, this is becoming your central nervous system. And if you look at the peripheral, so these are nerves. So they are supplying you throughout the body. So this is what? Peripheral nervous system. So if you look at the autonomic, what are the two types? One is the sympathetic, other is the parasympathetic. So sympathetic, we have to talk about them. So let's continue. Any problem out so far? So we start with it's just an introduction lecture. We just touching each and every basic aspect of your the histology, embryology, anatomy, neurophysiology. So start with the nerve. So there are two types of cells. One is the actual cell, main functional cell. Other is the assistant cell, or you can say supporting cell. So the main functional cells of your kidney is nephron. Of nervous system is the neuron. So as you call the study of your kidney is called nephrology, study of your nervous system is the neurology. It is related to their basic cell, which is neuron. So their actual cells which are performing their role, and the glial cells are what? They are just supporting their function. All right. So you can see, if look at this picture, so what you can see, this is what? Cell body, and here is your axon, and these are what? These are your dendrites, and you can see these are glial cells. Glial cells, they are just supporting cell. So they are present in both in the central nervous system and in your peripheral nervous system. So there are billions and billions in 10 raised to power 12 their number of, if you look at the number of the cells. So but you look at the glial cells, they are, as compared to neuron, their number is more than the neuron cell. Glial cells are, if you look at, their 90% are the glial cell which are supporting cell. So If you look at this is the simple pictures of neuron, you can see these are what dendrites. So these branches you are coming. What is the function of dendrites? They receive the signal to everything. It is related to central nervous system. So the dendrites, they are taking signal from the peripheral to the central nervous system, receiving signal, and the peripheral, the axon, they are taking away. So if you look at the your receptor, so at the receptor level, so at the receptor level, what is present? Dendrites or axon? Dendrites. For what? Receiving signal. At the effector level, what is present? Axon. So axon, they take signal from the central nervous system and supply to the effector. While if you look at the receptor, there are dendrites which are taking signal and sending toward the central nervous system. So if you look at this is the cell body. Alright, so here are dendrites, here are the cell body, these are axon. So if you look at here, there is something. What is this? Myelin sheath. So there are cells. This is also glial cell. So this formation of this myelin sheath is the function of glial cell. As I mentioned already, there is supportive function. So they are not playing acute function; they are just supporting a function. So what are these points? Where node of Raynaud. So your the nerve is coming like this. So coming here, coming here, coming here, coming here. So jumping nerve. So that's why in this type of impulsion or the innervation, so the speed is high because there is no need to change potential at this level because here is myelin sheath. So only this part. So it's make time shorter. It makes time shorter as compared to change the potential of throughout the axon. That's clear. Now these are the axon terminal. So we are at the level of effector. Okay. So any problem? These are glial cells, different cells. So just remember the Schwann cells and the oligodendrocytes. They are actually involved in the formation of your myelin sheath. Schwann cells in the PNS, peripheral nervous system, and in the central nervous system, the oligodendrocytes are important. If you look at these are macroglial cells, which are the macrophages, and the ependymal cells, they are actually lining. They form the lining of your central nervous system. If you look at these are what? Astrocyte, very important cell. So this is what? Artery, capillary. So here is the neuron. So if you look at here is neuron, here is capillary. What is in between them? Astrocyte. So they are actually creating the blood-brain barrier. Here is blood, here is the brain, and this is creating actually part of barrier. So one part is the inner layer of your capillaries, and then these food process, you can see these food process astrocyte are becoming part of the blood-brain barrier. So in this way, so the content which is coming in the capillaries enter into neuron by this barrier. So everything cannot cross this blood-brain barrier. So that they provide some aspect to the protection, important role. Clear? So you can see here myelin sheath. If you they are just wrapping. So if you see these are axon fibers. So this is your myelin sheath which is wrapping this neuron. So if you look at the one is myelinated and unmyelinated. If you look at the peripheral nervous system, so you can see it is also unmyelinated, but in the peripheral nervous system, it's not completely unmyelinated. So you can see they are resting like this, but not complete myelination. In the central nervous system, maybe unmyelinated completely, but in the peripheral nervous system, where you can see they are resting on the myelin, but they are not completely wrapped by the myelin sheath. And other important aspect, you can see this nucleus. is always outside on the outer aspect. Can you see? This nucleus is on the outer aspect. They are not coming at the center as the myelination started. So this nucleus remain always outside. You can see on the outer aspect. So nucleus is always on the outer aspect. So we have to discuss in detail in the next lecture in the discussion of the histology, but just an overview. So this is table in which you can see the function. So the CNS cells. So only two cells, the neurolimocyte, or you can say Schwann cell, same thing, and the satellite. These two are PNS cells, glial cells, and other four are the what? They are the central nervous system cell. So important is myelin production. One is the Schwann cell, other is the oligodendroid. Any problem, Shufa? So the types of neuron. So different types of neuron depend upon the poles. What do we mean by pole? A point where something came out. So if you see how many points where the things are coming, one is here, one is here. So this is bipolar. If you see here, how many poles are there? One. So this is unipolar. But we call them the pseudo unipolar because they actually from the cell body is coming one pole, but then they divide into two. So that's why we call them the pseudo unipolar, although it's a unipolar type. So here what you can see 1, 2, 3, 4. So more than 3 is considered as a multipolar. So and these are what you can see the N-axonic in which there is no exon. Clear? So the signals are coming from the dendrites and reaching to the cell body, and then from cell body toward away from the cell body is what? Axon. Alright. So we can unipolar, bipolar, and the multipolar. So most of cells of our in the central nervous system are the multipolar cells. So this is some histological point. So you can see. Now come toward the central nervous system. We have covered the basic concept regarding the neuron. So easy. Yeah. So this part you can see is the major part. If you see when you see the brain, the one picture which come in front of you is that picture. So that is what the major part you are seeing is what. This is what. Although it's all cerebral, and this part you are seeing right here is the brainstem. Actually, brainstem has three parts. On the upper aspect is the midbrain, then pons. On the lower aspect is the medulla, and medulla continues with the spinal cord. Midbrain, pons, and the medulla oblongata, which continues with the spinal cord. On the back of that, you will see the cerebellum. So these here is cerebellum. Here is what your cerebral, and here is the brainstem, which has three parts: midbrain, pons, medulla oblongata. So outer part or you can say cerebral hemisphere or cerebral cortex. So cerebral, there is the most of the time we will say that the signal are going to the brain, signal coming from the brain. Where are they going? So this part, this cerebral, where you will see the parts which are the motor, which mean they are controlling, they are giving signal to the skeletal system to contract or relax. So same like the sensation are going there. So they are all the motor function or the sensory function, they are reaching at this point. So this is function of your cerebral. So and if you look at the cerebellum, their main function is the correction. So the accurate movement. For example, I want to pick this pen. So like this, if I am try to read this pen, although the motor function is coming from my cerebral. So if the cerebellum, there is a problem in the cerebellum, my hand can overshoot or undershoot. The accurate movement is controlled by the cerebellum. Concept clear what I am saying. So although my hand is moving, but the accurate reaching at this point is the function of the cerebellum. So if there is problem in the cerebellum, so eye hand may overshoot here or undershoot here. So that is the function of cerebellum. So these are the frontal, parietal, and occipital and temporal lobe of your the cerebellum. So actually, when you visited somewhere mountain areas, you have seen there is a mountain, there are bulging, and there are the valleys, depression. Same like here. So the bulging or the or here are called gyras or gyri, and the valleys you seen in the mountain areas. So those valleys are present here. We name them the sulcus. So depression are the sulcus, and the bulging are the gyri. So here I'm not going to discuss all the sulcus, all the gyri, major ones. So this is actually central circle which separate the frontal lobe from the parietal lobe, and the parieto-occipital separate the occipital and the parietal lobe, and the lateral sulcus which separate the temporal lobe from the upper one, the frontal and the parietal. So actually your frontal lobe is concerned with the sensory areas, and your parietal is mainly motor function. So the frontal is with the motor function, and the parietal is with the sensory areas, and the occipital lobe is mainly concerned with the visual, and the temporal is hearing and speech area. So we have to discuss the cerebrum in later. So you can see here this is longitudinal feature between two cerebral hemisphere, and here is the sulcus, and here is what bulges gyrus or gyri. So just recall that this is what cerebral hemisphere, and epithalamus, thalamus, subthalamus, hypothalamus, midbrain, and here is what pons and medulla oblongata and cerebellum. So I'm just going to in the embryological. So how many layers are embryological layer? Ectoderm, mesoderm, and endoderm. Our concern in the development of the ectoderm mainly. So and little bit the mesoderm. So ectoderm is actually their bulging like so their folding occurs in the ectoderm. So some part is going to develop into second, and the middle part, which we call them the neuroectoderm, which is concerned with the CNS development. So neuroectoderm form a neural plate, or you can see here. So they are dipping down and forming a tube-like structure. So in front, if you look in this way, so you see a oval shaped structure, but when you put in longitudinal, you see the vesicle. So there are some primary vesicle, there are some secondary vesicle. So primary, then go to secondary, and then go to the actual structure. So here is so you can see convergence, and so here is this is becoming neural tube, and this part is neural crust, green color you can see. So our concern is the neural tube and the neural crust. I am not going to detail in this lecture. We will discuss in the CNS development. Just remember what are the derivatives of this. So you can see here, what you can see, these are primary vesicles. So which is concerned with the forebrain, midbrain, and likewise your GAT, in which you find the foregut, midgut, and hindgut. So these are primary vesicle, prosencephalon or mesencephalon, and the remainder. So then they convert into these secondary vesicle. So telencephalon and diencephalon from prosencephalon, and midbrain there is no division of midbrain, so single midbrain continue. And rhombencephalon, hindbrain divide into the metencephalon, melencephalon, and the lower margin of this tube is what spinal cord. So you can see here the primary vesicle, 3 primary, 4 mid, spinal cord. In the step 1 exam, they are main concern with the derivatives of this. So their main concern with these vesicle. So when you see these vesicle, some are the primary vesicle and some are the secondary. So can you tell me which one is the second vesicle? Diencephalon. Secondary, this is first, secondary, second, third, fourth, and fifth. Easier way, telencephalon is going to your cerebral hemisphere, and the lower margin diencephalon, or you can say is going to thalamus, and you can follow the alphabet MES, MET, so then YCAM. So if you go into the alphabetic order, so same like here you can follow the mesencephalon, metencephalon and melencephalon. So mesencephalon is going to midbrain and meten is going to pons and the cerebellum, and melencephalon is going to medulla oblongata. Please it up. So this is, and on the other side, if you look at the ventricle, lateral ventricle and the third ventricle coming here, and the cerebral aqueduct, a ventricle part, and the fourth ventricle and the central canal are taking origin here. So this neural tube lower margin, I think below the fourth somates is going to the spinal cord. So major concern is what are the derivatives. So neural tube form the CNS neurons, so and the glial cells. So there are mainly the ependymal cell, oligodendrocyte and astrocyte, while the neural crest cell, they are concerned with the PNS neurons and the PNS glial cell. So mainly the melencephalon and Schwann cell and the melanocyte, so they are coming from origin from PNS. And the last part, so two things are missing here. One is the cell of your glial cell of your neural tube, CNS. Which one? Which is four are mentioned here: ependymal, oligodendrocyte, astrocyte. One is missing, that is macroglia. So they are taking origin from the mesoderm. And out of three meningeal layer, your pia mater arachnoid take origin from the neural crust, while the mesoderm give rise to dura mater. So just
it's 7:46 p.m.
Microglia and alphabet D related to your D. So just for trick. So you can go through this and you can see here. So this part is coming from forebrain, this is your midbrain, and here is hindbrain. So this is the description of that. I'm not going to. So
Sorry,
we
I didn't understand.
and we continue the later part.